Hi, Munchkins. So these are going to be your notes over the cell cycle and division. If I ever talk too fast and you need more time to fill out your notes on a slide, please just raise your hand so that Mr. Adams, your sub, can pause the video and you guys have time to fill out the notes because you know me, sometimes I talk too fast. So let me know if you guys need anything. All right, so here's your guys' learning goals for the cell division and cell cycle notes is that you can describe how cells reproduce. And so that's what we're going to call the cell cycle. You can explain the products of mitosis and justify why it's needed. And then you can make connections between the cell cycle and other processes that we've learned so far, like protein synthesis and DNA replication. So let's talk about what the heck is the cell cycle. So you guys are going to focus on your guided notes on filling what is in red. So the cell cycle has some basics that are around it. So all organisms need to grow. Just like you guys have grown up since you were a baby, all living things have to grow. All new cells are produced from already existing cells. So we talked about it last week in class, right? We talked about the fact that your cells need to repair. Sometimes they're damaged and they need to be replaced, or sometimes they're old. And so if cells ever need to be replaced because of damage or age, they get created from already existing cells. And then some types of cells are continuously replaced. So your skin cells are cells that are continuously replaced because there's a lot of damage. Cells in the lining of your digestive system, because food moves through it and there's a lot of chemicals in it, are going to need to be replaced too. But then there are some types of cells that you guys saw in your activity today, the lifespan of cells, that don't get replaced once they reach maturity. So it depends. And the reason that cells are small and that you are made up of so many of them is because it helps them be more efficient. It helps it so that they are better able to regulate themselves and use energy well. So the cell cycle, when we talk about it, is an ordered sequence of events that take place in the cell and result in both cell growth and division. There are going to be two main parts of the cell cycle that we are going to talk about. The first one is called interphase. And then this is when the cell grows, the DNA replicates. Remember, we just talked about DNA replication last week. And it is also going to prepare for mitosis. And mitosis and cytokinesis are the two other parts of the cell cycle. And those are referred to as division of the nucleus and the cell. So this is a great diagram that shows the cell cycle. And so if you guys look, interphase is all of this blue stuff. You do not need to know how interphase is then divided into three different stages. But if you guys are curious, the three stages are G1, S, and G2. During interphase, when the DNA replicates, since you guys know about that, that actually happens inside the S part of interphase. And then mitosis and cytokinesis, when the cell actually divides, is just this part in the silver. So based on this diagram, what phase do you think the cell spends most of its time in? So I'm going to give you guys a second to think about that. So hopefully you guys said interphase, because if you notice, most of this is blue, a small portion is brown, and an even smaller portion is purple for cytokinesis. So most of the time, your cell is just growing, replicating its DNA, and preparing for my four-cell division if it is able to divide. And so there are some checkpoints here that we'll talk about in more detail later to make sure that everything is right in the cell during interphase as well. All right, so let's talk about interphase in a little bit more detail. So the purpose of interphase is for the cell to grow, to replicate the DNA, and for the cell to check DNA for errors. And this is going to be really important because we've talked about what mutations are. And a lot of mutations can happen during this time when the DNA replicates. And so after it replicates, there are specific things that it'll go through to check and make sure that there are no errors, and then to fix their errors if something wrong happened during DNA replication. Sometimes back in the day, they used to call this the resting phase of the cell, but the cell's not resting. This is when it's growing and preparing to divide. And like we said, this is where the cell spends most of its time. And some cells will exit the cell cycle during the beginning of interphase if they no longer divide. So like you guys saw in your activity, the lifespan of cells that you just did, there are various age ranges and if a cell can divide or not. So neurons, heart cells, muscle cells, all of those, or heart muscle cells, sorry, are things that cannot divide once they reach maturity. And so that's why things like spinal cord injuries and heart attacks are so can be so damaging because those cells cannot replace themselves. So that's why spinal cord injury, people who have a spinal cord injury may never be able to walk again because those neurons that make up your spinal cord cannot repair themselves and they can't grow and divide like our skin cells can. So again, like if you guys ever need more time to write something down, make sure you just raise your hand so that you guys can pause the video and then finish it in a second. But remember, you're focusing on writing what's in the red in your notes, and it's just fill in the blank. So during the cell division, the chromosomes this is when the DNA will compact into chromosomes. And so remember, chromosomes are how we store the DNA. It's the DNA wrapped tightly around proteins. And like we've talked about before, different organisms have different numbers of chromosomes. But in that organism, every cell has the same number of chromosomes. We will learn except for the sex cells, the sperm or egg cells. And so before mitosis can happen, before the cell divides, those chromosomes have to be copied because we want to make sure that every new cell has the same original set of instructions so that it can keep doing the job that it needs to do. This picture down here in the lower left corner, this is called a karyotype. A karyotype is a picture of the chromosomes of that individual. So it shows the 23 pairs or 46 chromosomes total for that person. The last set of chromosomes down here are the sex chromosomes. So this person has an XY set, so that means they're male. This is a way that we can tell if someone has chromosomal disorders by looking at the pictures, a karyotype of their chromosomes. So just to review DNA replication, I know you guys went over the answers at the start of class, but we need to talk about how the DNA replicates during interphase and make sure you guys understand the overall steps. So DNA replication is when two identical DNA molecules are made from one original one. And so you guys know that the DNA opens up, and the purpose of the DNA replication is to make a new set of DNA for the new cell. And so some of you guys might have seen in some of the readings that we did last week is that DNA replication is semi-conservative because there's one original DNA molecule, and then one of the strands is used as a template, and the other strand is used as another template that is eventually going to lead to the new DNA molecule. So one half of the DNA molecule is new, and the other half is from the original. And so that's why it's called semi-conservative. And here's a nice picture for you guys to look at the steps. So the DNA helicase opens up the DNA molecule, and then the DNA polymerase reads the orders of the A's, T's, G's, and C's and adds the correct nucleotides to make the other half of our DNA molecule. And so here's the steps that you guys need to have in your notes. You need to remember that the first step of DNA replication is that the DNA unzips. And then the second step is that a new complementary strand is made from the original strand of DNA by adding the matching bases. So by adding the complementary A's, T's, G's, and C's. So if you had an A from the original strand, that means that a T is going to be added in our new DNA molecule so that we still have those complementary base pairs. A and T still pair, G and C still pair. So it follows those rules as it builds the new molecule. And then the third step is the entire process continues until the entire DNA molecule is copied, which is so a lot, which is why we saw in the video last week that it had to go so fast because that's a lot of copying that it has to do. Remember, in every single one of your cells, you have about 3 feet of DNA. So that's a whole lot of A's, T's, G's, and C's that we have to copy before the cell divides. And once it's copied in the cell divide, each set of the DNA will go into the new cell so that those cells have these same original directions as before. So I want you guys to think about it. Pause the video if you need to. But how is DNA replication similar to transcription, and how is it different? So one of the ways that DNA replication is similar to transcription is that the DNA molecule has to be opened up so that we can read the orders of the A's, T's, G's, and C's. But what's different is what we make. During transcription, we were making mRNA. We were making a copy of a specific part of the DNA, whereas DNA replication makes an exact copy of the DNA and makes a copy of all of it, whereas the mRNA was just a portion of the DNA. It was just a gene. Good. All right, so now that we've talked about interphase, which is where the cell spends most of its life, we now need to talk about mitosis. And this is how the cell is actually going to make a new cell. So the purpose of mitosis is to grow, because as we grow, we need more cells. The other reason that we need new cells is because there might be damage to a tissue. So if you have a cut or damage has occurred on the inside, your cells are going to need to make other copies to repair those damaged cells. And then also asexual reproduction. So cells produced are identical copies of the parent cell. So asexual reproduction is how bacteria reproduces. And so they are just making identical copies of themselves. So just review for your notes. When we look at this whole cell cycle, interphase is the first part. And it's when the cell grows and replicates its DNA. And this happens before mitosis. And now we'll talk about the cell division, aka mitosis, and the general steps that are happening in them. So this is a nice diagram to show you the phases that we're going to talk about of mitosis. You will not be responsible for remembering all of the individual steps, but you need to understand the overall process of mitosis. So in the first step, which is called prophase, the DNA condenses into chromosomes and the nucleus disappears. Because remember, we said DNA can never leave the nucleus. But during this step, the nucleus is going to disappear so that we can organize the DNA molecules that have been duplicated and get them into their new cells. So after prophase is metaphase, and that's when the chromosomes line up along the middle. This just ensures that the copies of the DNA are organized so that each cell gets the correct set of DNA so that they're identical copies of each other. The third phase is anaphase, and this is when the chromosomes, we call them sister chromatids at this point since they're copies of each other, are pulled apart to the poles of the cell. So when we see this, this is what it actually looks like, and it kind of looks like pieces of spaghetti getting pulled away from each other. The fourth phase of cell division is called telophase, and this is when each set of chromosomes are on opposite ends of the cell, and the nuclei are going to start to form around them. And the last part is cytokinesis. This is when two separate cells are formed by closing the cell membranes around them. And the biggest thing that I need you guys to remember with mitosis, like when we do cell division, when a cell goes through mitosis, is that the cells that are made at the end are identical to each other. And we want them to be that way, right? We don't need to make cells that are all different from each other, because then they're not going to be able to function the way that they need to and replace the old and damaged cells. So we want them to be identical after they go through mitosis. And that's our review and the end of the notes. So you guys should be able to answer why do organisms need to create new cells? So remember, it's for growth, to repair damage, and sometimes it's for asexual reproduction, like in bacteria. That is how they reproduce. The resulting product of mitosis is to make two identical copies of the cell. And then why do you think the cells spend most of their time in interphase? What I'm really looking for you guys to understand is that because it's when they grow, they're replicating DNA and checking for errors, all of those things take time. And we want to make sure that they are done right before they get to move on to the next part of the cell cycle. So if you guys need to go back and review anything, let me know. If you guys have questions, write them down in your notes and we will make sure to talk about them when I see you guys on Tuesday or Wednesday. Have a good day, guys.